Hi, today we're going to look at anaerobic respiration, which is a set of alternate pathways that an organism can use when oxygen levels are low. Organisms still need to be able to survive in low oxygen environments and still be able to produce ATP in those extreme circumstances, those extreme situations. And so anaerobic respiration is a strategy or method that enables organisms or cells to actually do that. Now, what's key in anaerobic respiration is what you see right here on the screen. Normally, in glycolysis, glucose will give up electrons to NAD. And that's a normal part of glycolysis. That's the way in which energy is extracted from the sugar, from the glucose. When NAD gets the electron or electrons from glucose, it becomes this molecule right here, NADH. All right. Now the thing about anaerobic respiration is cells have a limited supply of NADH that's around. And if the NADH gets used up, then glycolysis is going to stop, and therefore ATP production is also going to stop for the cell. So in order to do anaerobic respiration, there has to be some way for the cell to take that NADH molecule or molecules that it has made and recycle it, to recreate the NAD so that glycolysis can continue. So in anaerobic respiration, there's no oxygen, there's very little oxygen around, and that backs up all of cellular respiration. And the only thing that can actually function for ATP production for the cell is glycolysis. And so if that cell wants to survive, given the extreme circumstances and environment that it's found itself in, it needs to find a way to continue to do glycolysis. And ethanol fermentation, lactic acid fermentation are ways in which that happens. And it's all about taking that NADH that's made in glycolysis and turning it back into NAD so that NAD can continue to accept electrons from glucose molecules in glycolysis. So the way that the cell does that is the NADH will actually transfer its electron to another molecule. When that happens, the NADH, if I just get rid of that hydrogen there, the NADH becomes NAD again and can be used to further accept electrons from glucose, and glycolysis will continue to make ATP for the cell. All right. So the first type of anaerobic respiration that we're going to look at is called lactic acid fermentation. So in normal glycolysis, glucose will be changed, if you follow my cursor here, will be changed into pyruvate. And what we get out of that is 2 net ATP, which you see right there. You also get 2 NADHs net, or total, being made as well. All right. So the ATP here can be used right away for cellular work. Now normally the NADH would go to the electron transport chain and be processed for ATP production. But the problem is, in the low oxygen environment, the electron transport chain is shut down. It's not operating because there's no oxygen around. And so these NADH molecules actually don't have a value for ATP production. So the cell is, squeaking out a little bit of ATP out of glycolysis. It's not much, but it's better than nothing. And so what the cell needs to do is find a way to get this NADH turned back into NAD, like you see here by the pink arrow, turned back into NAD, so we can take another glucose molecule, turn it into more pyruvate, and continue to get a little bit of ATP out of it. So what the cell does is it takes the NADH and transfers electrons to the pyruvate, and in a chemical reaction changes the pyruvate into lactate. That enables these NADH molecules, again if you follow the pink arrow here, to be changed back into NAD. And then the NAD can then be reused again in glycolysis and get some ATP. So glucose converts NAD to NADH, we make some ATP, the glucose becomes pyruvate, the pyruvate accepts electrons from NADH and it becomes lactate. the NADH gets changed back into NAD, and then we start all over again. Now other cells will do another form of anaerobic respiration called ethanol fermentation, which you see right here. Very similar to the previous process, but there's some slight differences. Again, we're doing glycolysis. Glucose gets changed into pyruvate. We get some ATP being made, and the NAD gets changed into NADH. Now, there is again that problem. We have a limited supply of NADH inside the cell, and we need to be able to recycle it or turn it back into NAD so that we can continue to do glycolysis here. So what cells do, some cell types do, is change the pyruvate into acetaldehyde, and they do that by removing a carbon dioxide. Then the acetaldehyde will accept electrons from the NADH, and the acetaldehyde will be changed in a chemical reaction to ethanol. That means the NADH can be changed back into NAD, and we can start all over again. So again, real quick, glucose becomes pyruvate, NAD becomes NADH, and we get some ATP. That's glycolysis. But the pyruvate then gets changed into acetaldehyde by the removal of carbon dioxide. The acetaldehyde accepts electrons from NADH and gets changed into ethanol. The NADH gets recycled back into NAD, and then we start all over again. So all this is happening in low oxygen environments. Of course, the product of this form of anaerobic respiration is ethanol, and in lactic acid fermentation, the final product is going to be lactic acid. All right. So that's the basic biochemistry of those two forms of anaerobic respiration, and of course there's some applications of that which we'll deal with in a future video.